I'm Uta Francke. I'm a human geneticist, originally from Germany, as you can hear. And I'm an emeritus professor here at Stanford and also senior medical director at 23andMe.
So when people talk about variation in the human genome, we oftentimes talk about single nucleotide variants, which we'll come to in later lessons for our course. Is that the main kind of variation that exists in the human genome, or these single letter differences, or are there other kinds of variation that are important too?
We have identified about 15 million single letter differences. So by sheer number, they are the most common. But then there are other variants, they are called copy number variations, where whole chunks of the genome can be repeated, and they can be present in either 2, 3, or many copies. And there are differences between human beings. So some people have 2 copies, some have 5, some may have 12. And if you look at the total number of nucleotides involved in those, then this is more common. So the individual copy number variants, or CNVs, are fewer than the ones we have identified for SNVs. But if you look at the amount of genomic sequence involved in this type of what's called structural variation, it involves a larger part of the genome.
I see. So between single nucleotide variants and copy number variation, those are the two main kinds, or the two main forms of variation in humans.
Yeah, then there are others like inversions, where certain pieces of the genome can be turned around in some people and not in others.
And even translocations. How viable are translocations, and do they get propagated at all in a population over time, or are most translocations inviable?
Well, we find them in families. There are balanced translocations, where a piece of a chromosome is taken off and hooked somewhere else, or 2 pieces have been exchanged. And as long as they are balanced, meaning that the right number of genes is there, people are fine. They wouldn't know about it, unless the break is in a gene, but that's pretty unusual. The break is most often in a repetitive sequence. But then, when these people may have children, because of what happens in meiosis, that the chromosomes align and cross over, it's possible that a child inherits a chromosome that only has an extra copy and is then gets the other one that's missing something. Then you have an unbalanced translocation. And that can cause very bad developmental defects, intellectual deficits. These are the children who we see in the clinic, and we look at their chromosomes and look at their parents, and we see the translocation has been passed on through several generations already. But otherwise, they don't last in populations.